This video will discuss neurotransmitters. Neurotransmitters must be synthesized in the presynaptic neuron, localized to vesicles, and released under physiological conditions. They must be rapidly removed from the synaptic cleft, have a receptor on the postsynaptic neuron, and elicit a biological response when they bind to the receptor. Neurotransmitter action involves 7 key steps. These include synthesis, storage in vesicles, leakage and destruction by enzymes, release into the synapse due to action potentials, binding to autoreceptors and postsynaptic receptors, and deactivation through reuptake or enzymatic degradation. Neurotransmitters can be classified as either excitatory or inhibitory. Excitatory neurotransmitters include acetylcholine, aspartate, dopamine, histamine, norepinephrine, epinephrine, glutamate, and serotonin. Inhibitory neurotransmitters include GABA and glycine. Acetylcholine is synthesized from choline in cholinergic neurons, catalyzed by choline-acetyltransferase. Once synthesized, acetylcholine is stored within synaptic vesicles. The release of acetylcholine is triggered when an action potential causes voltage-gated calcium channels to open, allowing calcium ions into the axon bulb. This increase in calcium concentration causes synaptic vesicles containing acetylcholine to fuse with the axonal membrane, releasing their contents into the synaptic cleft. Acetylcholine diffuses to specific cholinergic receptors on the postsynaptic membrane. Binding of acetylcholine opens ion channels, initiating an action potential in the next axon. Acetylcholine receptors are ion channel receptors made of multiple subunits. To prepare the synapse for another impulse, neurotransmitters are hydrolyzed by the enzyme acetylcholinesterase in the synaptic cleft. This produces choline, which poorly binds to acetylcholine receptors. Additionally, empty synaptic vesicles are returned to the terminal bulb by endocytosis to be refilled with acetylcholine. Acetylcholinesterase, or ACE, hydrolyzes acetylcholine. Its active site has 2 critical subsites, the anionic site for binding acetylcholine and the esteratic subsite where the ester bond breaks. This releases acetate and choline the choline is quickly taken up by the presynaptic membrane. Catecholamine synthesis includes dopamine, norepinephrine, and epinephrine. The first step is hydroxylation of tyrosine, catalyzed by tyrosine hydroxylase, which produces dopia. The second step is decarboxylation, catalyzed by dopa decarboxylase, which converts dopa to dopamine. Deficiency of this enzyme can cause Parkinson's disease. The third step in catecholamine synthesis is hydroxylation, where dopamine is converted to norepinephrine by dopamine beta hydroxylase. In noradrenergic cells, this is the final product. The fourth step, methylation, is catalyzed by phenylethanolamine N-methyltransferase, converting norepinephrine and adu-met to epinephrine and adu-HCY. This diagram illustrates the multi-step pathway of catecholamine synthesis, including the 3 final products, dopamine, noradrenaline, and adrenaline, and the key enzymes involved in each conversion. Serotonin is synthesized from the amino acid tryptophan through 2 reactions. The first is hydroxylation, producing 5-hydroxytryptophan, catalyzed by tryptophan hydroxylase. The second is decarboxylation, producing serotonin, catalyzed by hydroxytryptophan decarboxylase. Serotonin is synthesized in the CNS and chromaffin cells. This slide visually represents the 2 main steps of serotonin synthesis, starting from tryptophan and resulting in serotonin, catalyzed by tryptophan 5-hydroxylase and hydroxytryptophan decarboxylase. Serotonin is degraded in 2 reactions. The first is oxidation, catalyzed by monoamine oxidase. The second is dehydrogenation, catalyzed by aldehyde dehydrogenase. This results in 5-hydroxyindoleacetic acid. Other neurotransmitters include histamine, derived from histidine and catalyzed by histidine decarboxylase. GABA, or gamma-aminobutyrate, is derived from glutamate, catalyzed by glutamate decarboxylase. Nitric oxide is derived from arginine, catalyzed by nitric oxide synthase. This is a summary of the neurotransmitter molecules, their derivation, and their site of synthesis. For instance, acetylcholine is derived from choline and synthesized in the CNS and parasympathetic nerves. Serotonin is derived from tryptophan and synthesized in the CNS, chromaffin cells, and enteric cells.